Hello, everybody. I'm really glad to be able to participate in this year's AIC meeting, and I congratulate the organizers for being able to organize this after all. Let's talk about the characterization of bioactive volatile-based products and its efficiency in aphid populations in a pepper crop. This research has been done at IMIDA in Murcia, Spain. Some of you have visited the place, and we organized one of the EIC meetings back in 2005. This time I am accompanied with two colleagues, Manuel Cantó, that is presenting another oral communication, and Felix Martin with a poster presentation. The work has also been done in collaboration with Gloria Villora from the University of Murcia and Pedro Guirao from Universidad Miguel Hernández. The outline of the communication is present a brief background, then our research line, describe the experimental, and then a detailed description of the results, and brief description of the discussion and conclusions. The Farm to Fork strategy of the European Union aims at reducing chemical pesticides, nutrients, antimicrobials, and to promote organic farming. In particular, the first target to reduce pesticides is in the way to promote greater use of low-risk pesticides in which botanical extracts have an important role to play. Aphids have emerged as important pests in recent years for pepper grown under greenhouse conditions. The emitters belong mainly to the species Mythus persife, Aphis gossypi, Macrosiphum euphorbiae, and Aula cortum solani. They cause direct damages on the crop and indirect damages by virus transmission. We have developed a research line on the use of essential oils as botanical active substances. Previously, we have selected 5 products for having repellent or insecticidal properties against the aphid. We developed a product based on the mix of these 5 compounds in equal amounts. The 5 compounds are namely citral, the blend of 2 isomers, transanetol, farnesol, cisjasmone, and lemon essential oil that contains limonene. Essential oils are produced by condimentary plants, and also they can be obtained from waste streams. The experimental included nanoemulsion preparation with the products, nanoemulsion characterization, testing the aphicidal activity in laboratory, and also in field experiments for 2 years. For preparation of the nanoemulsion, we use a high-speed rotor by mixing the oil in water at a percentage of 1% of the total bioactive volatiles. Formulations were developed using synthetic twin 80 or natural soya lecithin and sunflower oil surfactants. For characterization, we use a synthesizer that measures particle size and particle size distribution, and also theta potential. To test the stability of the samples, we also use an ultrasounds. To test the activity in the laboratory, we use a computer-controlled spraying apparatus and petri dishes with pepper leaves with settled aphids. 20 replications were used per treatment, and the mortality was recorded after 1 or 4 days. And results were analyzed statistically by various tests. Experiments were done at Torreblanca Experimental Station in Campo de Cartagena in Murcia for 2 years. In both cases, 2 greenhouses were used. During the first year, we had the experiments during spring, and in the second year, during winter and spring. In the first year, we evaluated the 3 treatments I have already mentioned with the 3 formulations. And then in the third year, we selected one of these formulations and then included another 2 treatments, one with a vegetable oil and another with a single product alone. In all cases, we recorded number of alive and dead insects after 1 or 4 days and evaluated statistically the results by non-parametric tests and ANOVA. Here we have some of the results. With the petri dishes, we obtain efficacies of 45, 71, and 63% of the formulations 1,2, 1,1, and 1,1,1. Therefore, we see that concerning the reduction of aphid population, the formulation with soybean was the more effective, and there was a clear difference with a less significant difference at 5%, comparing the formulation 1,2 with twin and the formulation 1,1 with soybean. Here we can see a result of the field experiment. Here we obtain that the formulation with soybean was the most effective in reducing the number of alive aphids. This was also obtained after 1 day and after 4 days of the treatment. In the two graphs in the upper side, we see that there was a difference between treatment and control. Also, there was a reduction in number of dead aphids, but in this case, there was a significant difference for the formulation 1,2 with twin 80. In the box and whisker plots of the that we can see in the lower graphs, we see a difference between control and treatments, also for first day and the 4th day. Aphid mortality was evident in all formulations, but was more clear with the formulation with twin. Some phytotoxicity was observed for the formulations 1,2 and 1,1. This was seen as a dry tips in the leaves and also a round dry mottle in the leaves, but there was not effect on the fruits. For this reason, there was the selection of the formulation 1,1,1 that contains sunflower oil, in which the symptoms of phytotoxicity were less evident. Let's move now to the characterization of the nanoemulsions. Here we can see the mixture product at 1% in the formulation 1,1,1, the one that was selected. In the red line, we can see that there are peaks of population, particle populations at around 500 nanometers and 150 nanometers, while in the control we have a main population at around 250. In both cases, there were larger particles, but in less amount. In this table, we can see a summary of the characterization data comparing formulation 1,2 with twin and 1,1,1 with soybean and sunflower oil. We see that when we use natural surfactants in the formulation 1,1,1, particle size is greater. Here we see a range from 216 up to 439 nanometers, while in the formulation 1,2, the range of particles was from 10 to 341. However, there was more stability in the particles in the formulation 1,1,1. We can see this in the zeta potential values that were minus 28 millivolts. Zeta average values were reduced from 250 nanometers to 100 nanometers, and they were stable in time if we apply ultrasounds. We also observed that cis-hasmone, when formulated with twin at a concentration of 0.25%, gave a very fine nanoemulsion with particles around 15 nanometers and a polydispersity index less than 0.3. These values were better than those with the formulation 1,1,1 that had particles with 353 nanometers and a PDA of 0.48. Therefore, we selected also this formulation for the experiments, the field experiments in the last year. Therefore, in the field experiment of the last year, we had the two products selected, the mixture product 1,1,1, the cis-hasmone product with the 1,2 formulation, and we included the third treatment with a vegetable oil, cottonseed oil. The experiments were done in greenhouses like this. The plants were inoculated with the aphids prior to the experiment. Then we apply the sequential treatments First, the mixture product, second, the cottonseed oil, and third, the Tiscasmon in the same plots. And we can see here the results of the number of alive aphids throughout the experiments. In the first case, in February, in the second case, during April. We can see that the treatment maintained the population with lower values during winter throughout all the experiment. The arrows represent the different treatments applied. However, in spring, there was a reduction in the second and third treatments, but in the first treatment, the effect was much lower. However, there were statistically significant differences in the instantaneous rate of population growth of the aphids and in the aphid mortality for the treatment with cottonseed oil, according to Wilcoxon test, and also in the treatment of Tiscasmon for aphid mortality, according to the same test. In spring, there was a difference, a statistically significant difference for aphid mortality in the treatment with Tiscasmon. Here we can see a summary of the statistical differences with the control of each of the products. There was not an effect in the case of the first treatment. There was statistically significant differences in the second and in the third treatments, but with some variability. Here we can see the aphid mortality that was obtained after spraying with cottonseed oil and soap that gave the best results in the experiment of this year. When we see the data of aphid mortality, we can see that during winter, we obtain over 80% mortality of aphids with cottonseed oil and Tiscasmon. During April, the results of mortality were much lower, but nevertheless, they were significant for the case of cottonseed oil. By selection of the formulation 111 of the mixture product, and by reducing the dose to from 1% to 0.5%, and also by application of the product by spraying to the air instead of the plant, we obtain less phytotoxic effects. However, there was also less effect on the pest in the case of the product we have developed with a mixture of compounds. Therefore, more research is needed on the formulation and method of application of the bioactive volatile product to produce better results in terms of aphid control and to get rid of the phytotoxic side effect being observed in the formulations tested. And with this, I thank you very much for your attention.